All right, so we all know and appreciate Sushma Naithani, Dr. Sushma Naithani, as one of the newer members of the Ag Biodata Steering Committee, but she also has a lot of other roles and activities. So research focuses on how using systems level pathway modeling to understand information flow in living systems and how evolution shapes this flow. And as part of that research focus, she is also really relies on and generates high quality biocuration information, gene orthology-based predictions and analysis of omics data. So sort of exactly what we like to talk about and promote here at Ag Biodata. As part of this, she serves as Senior Curator for Plant Reactome Knowledge Base, and she is the Outreach Coordinator for the Green Project. She does a lot of other outreach as well. So she is currently on the Executive Committee for the International Society for Biocuration and is the chair of the awards committee in that organization. She is also on editorial boards and is, of many journals and is the editor-in-chief of Current Plant Biology. So there's many more other things that she does, but I'll stick with that and I will turn things over to you, Sushma. Thank you so much for giving this webinar on the Plant Reactome resource. And you're muted, Sushma.
Hello, everyone, and good morning, good evening, or good afternoon, wherever you are. So I'm going to share my screen and talk to you a little bit about Plant Reactome. Okay. So many of you are familiar with Plant Reactome. Plant Reactome is a pathway portal of Grameen database. Most of the Plant Reactome biocuration happens at Oregon State. So if you have any questions or you like to reach out to us or to me, you can email me. Here is my email address. Little bit about how do you access Plant Reactome if you haven't used it. So you can go to Grameen webpage, and in Grameen webpage, actually you can search for genes or proteins or pathways or small molecules, metabolites, and you can reach to Plant Reactome portal. Or you can click here on Plant Reactome and also then you can reach into Plant Reactome portal. So a little bit about the history of Plant Reactome. And so Grameen project, as most of us and here know that it started in 2000, and but the pathways were incorporated in 2008, and we started with the biosite-based platform, created rice site, maize site, brachia site, sorghum site, some of the white site, fragaria site. So some of the 6 to 8 metabolic networks that were species specific. And then in 2012, they adopted another platform that was Plant Reactome, and it got started and it was funded by NSF at that point. So Plant Reactome has received funding only one term, that was in 2012 autumn, and then our funding from NSF ended by 2020. And now we have some support for Plant Reactome from USDA and also from NASA. So over the years, a little bit about the evolution of Plant Reactome. So of course, we had those rice cycle. We used rice as a reference species. And so we started with putting the pathways that we have created for rice cycle. And first we started with metabolic pathways and transport pathways. Soon then we started adding hormone signaling pathways, hormone biosynthesis pathways, and connecting them. Then we thought of adding some of the gene regulatory interactions in these pathways, and we created gene-gene interactions and tried to use the Reactome data model to create these pathways. And then we started looking into what else we can do. And so we thought of we can think about plant development, organ formation, organ differentiation, also these biological processes as a pathway. And we started curating plant developmental processes and also biotic and abiotic stress response. Then the idea was that all this curation that we were doing, mostly based on the published literature in rice, and sometimes we have referenced also the literature that is available in Arabidopsis. And so the other thing was that how we use the omics data to use for biocuration of genes and pathways. So we started looking at more recently in past 3 years. And so as you see that most of the omics data that is available is transcriptome data. And so we looked into transcriptome data and how we can use that for creating transcriptional networks, adding gene regulatory information into our existing pathways. And now with a little funding from NASA, we are also looking into gravitropism pathways, how plants respond to gravity and microgravity environment. So this is a little bit about how plant reactome has evolved and what it offers. So you can go to plant reactome website also and access plant reactome. And here is the web address for plant reactome. So currently we curate in rice, but based on orthology projections, we are able to project the pathways for 120 species, ranging from unicellular cyanobacteria to higher plants, monocots, dicots, and you can find species of your interest in this database. This is just a kind of the progress of how we added many species, started with rice, but then soon we added grasses and then other model organisms from the plant kingdom, and then soon many other species that have evolutionary importance. And as the genome sequences became available, as the transcriptome sequences became available, our species number grew, and currently it is 120. Similarly, this is the content of the number of the pathways that we increased. And so currently we have about 326 curated reference rice pathways that are projected to 120 species. So then, you know, that this is an exponential if we add curated and also the projected pathways. And again, here you can search for in plant reactome homepage for gene, protein, metabolite, pathway, and so on. So for browsing, one can go and hit the pathway browser, and we use the fireworks view just to provide an overall view of the pathways that we have in different categories. As you can see, we have metabolism and regulation, that is the majority of the pathways, but we have some of the pathways around the circadian rhythms. We have some growth and development. We have response to biotic and abiotic, and some of the conserved cellular processes. So all these pathways are organized hierarchically based on enzyme ontology and, you know, how different pathways are related. And so one can click on and, you know, get into the reaction level detail that I will show. So here are these, all the arrangement of the pathways. And then, you know, using some of these panels, you can close one window or, you know, open one window or two windows and so on. So here is a list of pathways on the left panel. Here is the main pathway diagram on the right top. And in the bottom, we display different type of linked information. So that information could be pathway description. It is like about the molecules, structure, expression, analysis, and so on. And through these links, we are able to connect to Ensemble, Unitrot, PubChem, CABI, Expression Atlas, and so all those that were the roots or the places from where we take our data, we collaborate, and we, you know, work with those other databases. So by default, we display Oryza sativa as a species here, but you know, you can choose any species out of the 120 species that are right now in the plant reactome. So what we can do, so of course, you know, we have also analysis tools for the users. So what users can do, they can browse and they can search pathways, but they can also upload their omic data. So it could be gene expression data, transcriptomic data, metabolomic data, proteomic data, and they can see, do the enrichment analysis and analyze their data, visualize their data in the context of the pathways. So this is just an expression of transcriptome data upload. And as you can see, genes that are upregulated are towards the red color, down-regulated towards the green color. And then when you click on any particular enzyme that is listed here, what you can see that you can see the number of the genes that are coding for the similar enzyme or the protein. So you see homologous genes, and then you can see how these genes are expressed in the samples you have provided. So as we know that plants have large gene families, they have multiple members of the same gene family, which bear the similar Go annotation. So this kind of analysis actually can help users to differentiate which of the homologue is playing a role, say, for example, in leaf or under stress or in fruits or roots and so on, and help them to shortlist candidate genes. So, and you can download these results in the Excel file. You can go from one pathway to another pathway and visualize your data. The other thing users can do also, they can do the species comparison, how the rice reference pathways stand out for Arabidopsis, maize, or any other moss or any algae, and which are the conserved pathways and which are the pathways for which we are lacking genes. We can also see the gene orthology relationship. So when you have the same number of genes in rice and projected species, the box is completely green. When you have the difference in the number of the orthologs in one species versus another, then you see half green and half blue box. So it can also provide some of the insights into the evolution of a particular gene or gene families. We also bring in expression data, that is gene expression, basal gene expression data from different tissues. And if you have a pathway, you highlight a particular gene here or gene product here, then you can see how that gene is expressed and where it is expressed, and that data can be displayed. For the differential gene regulation, one can access the EBI plant expression atlas and connect with the gene and to the data set. So we also facilitate that. We understand that our biocuration approach is very slow. We have very few resources. So what we do, we also try to use how other curated or other type of the omics data. So one of the first data that we started using was the gene-gene interaction data. So now we have gene-gene interaction data not only from rice, but also from many other species. And this way, our biocuration doesn't limit use of this data to only rice. You can apply this to any species. So what users can do, if they have gene-gene interaction data set, they can upload that to our pathways. And if you find interactor, then this number is, 2 here and 2 here in those nodes that plant reactome offers, or 10 or whatever, then you see the connections with that data. And then here is an example of interaction data from Arabidopsis, and then it identified a node in plant reactome, and it has, for example, 48 interactor. And that data can be displayed in this wheel card way, and you can use that how the genes that are not currently in plant reactome can be associated with some of the pathways or some of the reactions. So this is really nice because, it somehow complements our slow biocuration and allow us to look into some of the genes in interaction data and use them. As a researcher, one can have the more data-driven hypothesis and have more nodes available to them. So this is one example of how we are using omics data with plant reactome. The other type of the omics data, or the large-scale high-throughput data that we are trying to use in plant reactome, is to improve in gene functional annotation information and to think about which genes are expressed where and where those protein products are localized. So, for example, here is an example of one of the oxidases that are involved with ROS pathways. And so there are 3 members that are expressed in different places. So in plasma membrane, in plastic in mitochondria, and they trigger some of the reactions. And so if we use the subcellular location information from literature and from some of the predicted databases like CropPal, it helps us to put these three different genes that are encoding the similar enzyme but located in different places to visualize differently and see how they work. So that is another way we improve our gene functional annotations and improve our pathway biocuration. We also use some of the ways how we can link biosynthesis to the transport reactions, and so use this eukaryotic architecture of plant cell to inform our users that where those reactions are happening and how they are being connected. So some of the translocations reactions and transport reactions and so on. So this is the second example how we use high throughput data to improve our biocuration. Now, as I mentioned before, that most of the data we have is the transcriptome data. And so how can we use the transcriptome data? So of course, we can project those expression data in plant reactome that I showed you before. And we have, a lot of data, for example, from rice that is a monocot model from different stages. And so one of the things that we could do is to look into large gene family members and really see evidence for where they are expressed, and then use that annotation and use that evidence to link genes where they are more relevant. So similarly, we have rice is a crop that grows in upland, that grows in the lowland, that is grown in the flooded areas, and then that is also in the coastal areas. And so there is a lot of data on different rice varieties under different type of these stress conditions. And so we have a lot of differential transcriptomic data. So we can also use this data in a similar manner, and the expression data then can help us to improve biocuration of the gene family members. So recently we published this paper about improvement into gene functional annotation of SDRLK gene family that has about 150 members, and how we can use transcription data to figure out where those genes are, because they have the similar, all the genes have almost similar Go annotation, so where to go from that. So then we use the expression data, transcriptomic data, to say this gene is induced by stress, this is expressed in a certain tissue, it has more ubiquitous expression, this gene. So we use that information to improve some of the gene annotations, and that can further be used linking with some of the signaling pathways that we are biocurating. So here is another example of the biocuration of signaling pathway and how it can connect to the various metabolic reactions and other pathways. So this is the pathway of the biotic stress response, how actually plants recognize a fungal pathogen. So that has the chitin as an outermost molecular signature that is recognized by plants, and then peptidoglycan from the bacteria, which are the receptors, and how that cascade is activated and how it translates into some of the transcriptional regulation as well as some of the activation of the metabolic regulation. So this is just one example from plant reactome. So we had actually very fortunate to have a lot of undergraduate student curators who participated into use of omics data and for improving gene functional annotations for genes that we are curating and some of the pathway curation. So during the pandemic, past 2 years, we had almost a virtual classroom of the students, and we met virtually, and a lot of data was curated virtually. And that way, we see that we had a little advantage over the people who are tied to the labs. And so I like to present some of the work that was done by undergraduate students. And so students actually worked on a lot of different abiotic and biotic response pathways. And so the strategy we used different type of omics data. So for example, I told you we use the transcriptome data And we also see how these genes are co-regulated and if they are in the similar co-expression network. So that was one type of information that we built on. And then it is possible that two or more genes, they are co-regulated, but not necessarily they have the same transcription regulator or they have any target or transcription factor relationship. So we thought maybe we can look into specifically on the transcription factors. All are located in the nucleus and they can act both as a target and they can also act as a regulator. So we shortlisted some of the transcription factors and then we looked into if they are in the same co-expression network. And then we conducted also promoter analysis. So promoter analysis then helped us to score their direct relationship as a TFM target. We also then so if we see the co-expression and we see the presence of the transcription factor binding site, then we say, okay, these two lines of evidence are enough to establish this relationship of direct target and TF. So similarly, some of this information, again, we used for improving gene functional annotation. And then, of course, the information that was available from experimental evidence from the literature, we marked that, say, for example, for sure, things have been shown experimentally that this transcription factor binds to the promoter of this. And, if they are in the transcription regulatory network, that was, again, a double. It gave us some more confidence. So we were able to curate this data with different confidence, and then we used also the type of data that is the clip PCR or clip sequence data about the TSS mapping, some of the sequence data. So together then we are using to curate some of the transcriptional network. So we are doing that for different abiotic stress, ranging from heat, salinity, cold, and heavy metal response, all different. So almost similar strategy for submergence. So all that work is done by undergraduate students. And then we have also some of the students who are more computationally skilled. And so they are looking into the back of the plant reactome data set, what we have for 120 species and which pathways are conserved, which pathways are not conserved, which are specific to rice and to the grasses. So at different level, we are looking into the data we have curated over past 10 years to learn about the evolutionary history of metabolic pathways and also other type of the pathways. It also provides some kind of the quality control check for our biocurated and gene ontology based projections. So how you can do different things with the Grameen, other tools, and also with the plant reactome. We have YouTube channel and you can access many tutorials here. You can also access the presentation made by our undergraduate students in short talks that they did. And finally, I like to acknowledge our team. So at OSU, I work with a lot of undergraduate students, mostly on biocuration. And these students are funded through various programs. So some of those are through the USDA AgriU summer internship, other students through the URSA fellowship by OSU. And then some of those students are taking some of our courses for the research credit like Botany 003, 401, 40, and honors thesis students. So through various mechanisms, these undergraduate students are funded. Recently, we received a NASA grant for biocuration of gravitropic pathways, and current funding is also provided from the USDA and with the support from Doreen Group at CS Cold Spring Harbor Laboratory. Previously, Grameen has been funded by NSF and also from the USDA. I like to acknowledge also Human Reactome Database team for their support and also for the review of plant reactome pathways. So Human Reactome is our big collaborator, but then as I listed in our first slide, we collaborate with almost all public databases. We are very thankful to our users and collaborators. So if you like to take a survey of Grameen, then here is a link. We will appreciate if you fill that survey and we learn from you what you need from Grameen. And also, if you like to access any of the outreach resources, here is a link for that. We have also mailing list that you can subscribe to, and you can follow us on Facebook or on Twitter. And thank you. I'll be happy to answer any questions you have. Hi, Fiona.
Hi, that was a fantastic talk, and it's really, really impressive to see all the work you've done. I missed it when you mentioned it, but you did mention the ontology you were using for your pathways. What was that, please?
So we use the enzyme ontology to hierarchically how we organize our path, our schema of the pathways. So that is one place we use ontology. We use also ontology, plant trait ontology, and all different type of ontology when we write the gene summaries, when we write the pathway summaries, and how we curate genes. So we always use all of the Go gene ontology. So we have the slots in the plant reactome schema to use ontology.
And if I sorry, if I could just follow up with a quick question. When you've used this and you've taken from rice across to the other species, have you ever done an analysis to get a sense of how many genes, how complete, I guess, the pathways are when you transfer to other species?
Yeah, so we run that statistics, and with each release, then we provide that data for each species. If we use like 1,000 rice reactions, how they are projected in maize, and so that is always available in our release notes.
Okay, thank you.
Hi, Luna.
Hey, that was an awesome presentation. Thank you so much for that. So I have a couple of different questions, and I guess the first one sort of comes off of what Fiona asked with respect to other ontologies. So the first question is, are there terms or processes or pathways that you are finding that are not represented in the gene ontology, and therefore, we need to work on a better representation for those pathways or biological activities in the Go? And then the other question is how much synchronization there is between the pathways in Reactome and PMN. In other words, does every plant Reactome pathway have a corresponding PMN pathway ID? I guess what I'm trying to get at is sort of the interoperability of all of these different ways in which, pathways and enzymatic reactions and stuff like that are actually described.
So we don't directly collaborate with PMN. And I'm not, so I know that many species are common in PMN and also on plant reactome. There is a difference in how we curate pathway in psych, that is PMN is based on psych. So what they do, they use the genome sequence and then they run it against the predicted proteins, and they run into classification through the Go pipeline, and then they project and they predict. So they work from the top to down approach, and then they improve on or add the biocuration. What we do, our approach is different. We start from the base. We look into genes, we look into the proteins, we look into metabolites, and we curate those reactions with the support from the literature. So we don't use, so our is like the bottom up approach. And so that is like one big difference. The second difference between these two platforms is that, so therefore we don't have any corresponding IDs. Previously, we have used also rice psych, and we have curated many species-specific psychs that were very similar to PMN. And some of those we have done curation species by species and improved those. And so some of that curation we have brought into plant reactome from that. But still, when you do the projection using the psych platform, if you have Go annotation on 140 members and they are mapping to the same enzyme, that is there. And so you see like more representation and more association. So in plant reactome, we have actually cleaned that up. So if you have, say, you know, some of those kinases or, you know, the GSTs or some of the isomerases, and you have on every reaction where isomerization reaction is there, 100 or 50 of those mapped, we have cleaned those up. If there is no evidence for expression, if there is no evidence from protein, and you know, if some of these are pseudogenes and they are not coding, so we remove that, and maybe you see that in our database, maybe 3 or 4 or 10 are associated with that particular specific reaction. So our data is much more cleaned up in that sense. But then content-wise, perhaps we, you know, when you run the statistics of number of reactions or pathways or things, we might be low.
I guess the other, sorry, if I could just sort of follow up too, is because we at TERRA, we are primarily focused on using the gene ontology to describe gene functions. Yeah. And so occasionally, you know, I'll come across a paper where they describe a pathway that isn't represented in the Go, but then I would go and I would look to something like, you know, plant reactome or plant psych for, you know, ways of describing it and, you know, to sort of help go flesh that out. And I know that, like, you know, Reactome collaborates heavily with the Go in terms of, you know, modifying the structure. I guess what I'm sort of asking is, like, if you come across things that you feel should be added to the Go, then maybe we should talk and make sure that we're accurately representing plant biology in that as well.
Yeah. So we use all the Go annotation that is available, and when we curate, you know, the problem with the Go is that it is a very upper level curation, right? So it tells us only 3 categories, that is the cellular location, catalytic activity, and, you know, molecular component. Only 3, right? But then when you think about the pathways and the reactions, they are much diversified. You think about a tree taking a root from the Go and then branching out. And then not all the branches, you can of course correlate and go back to the Go, but the Go itself is not very sufficient to describe all the fruits and flowers and leaf, right?
So, no, that's not what it's intended to do. I'll follow up with you after this and just sort of, I can clarify.
As much as Go, and we, you know, include all the Go, but then we expand beyond the Go. And we add that whole biocuration that we are doing now, based on omic data analysis and everything. We are trying to complement Go to make the information more rich. Okay, anybody else? If I don't see anyone.
I have a question if nobody else. Oh, I see Cecilia unmuted herself. I'll let you go first.
Yeah, hello. Yeah, just one question. How do you assess the quality of information from literature? And do you associate actually the information to the pathway you build? Is it possible to have the source of information when you look into a specific pathway?
Yeah, so we write the summaries of the pathway. So every pathway has summary, and we have learned how to write better summaries over the years. So you will find that our recent summaries are better than the previous summaries. But then everything we write in the summary, every sentence has a citation from a paper, if it was coming from the paper. And so we use that evidence code also inbuilt in our things that is not publicly visible, but you know, we use author statement, experimental evidence, you know, the prediction. So we do have those evidence code that we associate with each reaction and each statement that we make. So you know, so all those things we have learned along the way, and we're trying to improve on it as much as possible. So you will find that information. We actually have to go back and actually do that for some of the pathways that were curated previously.
Thank you.
All right, I have a question, Sushma.
Yeah.
And I apologize, my computer crashed as it tends to do during ag biodata meetings, and I was out for maybe 5 to 10 minutes of your talk, so I'm sorry, I missed most of it. But, and if you address this, please let me know. But I was curious more, since in ag biodata, we're often concerned about how processes work and how we can improve those across databases and make, learn from what other databases have learned previously, so new databases don't make the same mistakes. And from what I gather, you do a ton of integration of data from various sources to make your plant reactome data so valuable. So in the years that you've been senior curator at Reactome, could you share any experiences that you had where you had to overcome data integration challenges and what those were and what we might be able to learn from what you've done at Plant Reactome in terms of combining data from different sources?
I think the biggest challenge is that we don't have enough resources and we don't have enough funding for the biocurators. For Plant Reactome, we had only one full-time biocurator. And, my position mostly was for the outreach, and a lot of biocuration that I did was voluntary. And then we found mechanism to involve undergraduate students that were available at OSU. So that was, I think, something other database folks can think about, involving undergraduates or graduate students and taking some of the biocuration practices to the classroom. And also, so I think it is double way, because it helps you to overcome the limit of the funded biocurator activity. But also it teaches the new generation how to use databases, how to have the information that is verified, how they can take the information from different experimental procedures and how to compare them. So teaching them skills. So when we actually do curation for Plant Reactome, we go to NCBI, we go to TIER, we go to REP, UniProt, and many, PropPal. So they get at least the informal education of how to browse things. So for all the databases that are relevant for genes and pathways. So I think that is something I like to, share, and I have a list of things, how I train my students. So if anybody wants to do that, I'll be happy to share my list and videos and things like that we have learned over the time. So that was one thing. In terms of the challenge, we still do not have mechanism to biocurate in species other than rice. And we don't even have funding, but if we get funding, we are hoping that we can have the vidi plantae as a reference rather than rice as a reference, and have some ability to curate in other species. Because, rice doesn't have all the metabolites tomato has, or, grape has, or something like that. So we like to have like more enriched curation of secondary metabolites. That is currently lacking. So that was the funding question, right? So, we still are struggling how to fund and get the funding for the Plant Reactome. So that was the second thing. And I think the third thing is that the harder thing in terms of biocuration is that linking all that pathway and genes, genotype data to the phenotype and to the images of mutants and different type of things. So still we don't have a repository, for example, that has a good curation of mutants from even Arabidopsis or rice. And, we have linked to the genes or group of the genes, how we visualize those simple gene mutants or the genes or the mutants that are result of multiple genes or epistasis and other type of genetic interaction. So our, hope is that someday we are able to link the phenotype. So the good part is that Plant Reactome at least gives you capability to link the genes and proteins and at least the biochemical phenotype that is measurable, not by eye, but with the technique to at least link that. But we are still not able to link mutant data. We provide the description and we refer to the papers. But, it will be very nice if you click on the gene and you also get a picture of the mutant or somehow have another link for that. So that is the challenge.
Lenore.
So thinking about that, for for resources like that do exist, like in Reactome, could you link out to example for like maize GDB for like to mutants associated with those genes? And
we haven't done that yet, and we don't have resources for that, but it will be really nice if we have, Arabidopsis mutants, we have rice mutants, we have maize GDB, and we are able to link it out. So we don't have right now any support to do that. And then there are a lot of reports in the literature, and those mutants are not in any database. In fact, there is no public repository for the mutants pictures and images, and how do you connect those with one gene or multiple genes?
Yeah, thanks. That was what I wanted to know. That's part of what we're trying to figure out here in AgBioData. Is what we know there's lots of progress, but lots of problems, not enough funding. What are the things that we can work together to solve? And maybe we can't absolutely solve funding, but we can try to make progress on that front and identify what can be solved first. I had another like super technical question just amongst us here. So for your software backend, is it your own software, or do you use a different platform that you add on to?
So we use the infrastructure and the central database of Reactome codes. And so whenever we
of what?
Of the human Reactome.
Okay, got it.
And so whenever we curate any pathway or anything, we put it in the central database and we share that with the human Reactome. And so we share small molecules like ATP and a lot of biochemicals and metabolites that are conserved between humans and plants. And so we don't have to create them again and again, right? And so we use their schema. If they improve on data visualization, we would benefit from that. So they are our great support.
Great, yeah. Does the human Reactome then also take your data and propagate it in the Reactome database as well? I mean, I know they do some orthology computations, I think, to project other species, or is that?
They haven't done, because they started in 2000 and we started in 2012. So actually it was the other way around. So for DNA replication, for some of the conserved cellular processes, actually we were taking their data and projecting on the cellular processes section, very limited data. And then slowly, as we are reading paper, we are replacing those with more curated pathways, because we find more detailed information in plants.
Can I add something? This is Pankaj. Yeah, they used to have Arabidopsis and rice for a long time, but when the plant Reactome became as its own entity at that time, the plant data was withdrawn from human Reactome, and so after that point, we are the major providers for that. So, but in terms of software stacks and everything, I think there's a lot of there's Justin Elser, when he's on the call, he may be able to tell more, because what we do is we, at the time of the production, we take the plant slice from the curated database, which is shared with the human Reactome folks, and then we do our own production on the same stack that the development version is on it. But it does have a lot of NoSQL stores and other things and indexing files to run through that. Absolutely. And another thing that we have is obviously the RESTful APIs, but there are also pathway widgets. If you have, and like we have already projected it for 120 species. So if somebody is running a genome for that particular species, the data can be pulled automatically based on the gene ID or the stable IDs that we provide. And then they can show the pathway, the whole hierarchy in their own pages and resources, and also use it remotely for doing any data analytics on their side. Thanks.
Great, thanks. It's a good way of reusing code and existing work. So. All right, are there any other questions for Sushma? Gone. Okay. Well, let's thank Sushma again for a wonderful talk, and this was a great discussion, everyone. So appreciate you taking the time, and Hopefully we'll see some or all of you at PAG, and then for our next webinar coming in February. So take care, everyone. Thank you.